So this is section 4 of chapter 8, and the topic that we're going to discuss is individual differences and variability. All right, so as we've been discussing, most studies are going to manipulate some independent variable, thereby creating at least two different treatment conditions. And then you're going to compare those two conditions against each other to look for significant differences. Now suppose you compare the average scores in condition 1 with the average scores in condition 2. And suppose you find some difference between those averages. And you will almost certainly find something. I mean, the chances that the two averages will be exactly identical are very, very small. So there's almost certainly going to be some difference between your two conditions. So the question you have to answer is whether this difference is significant or not. And in order to answer that question, you need to know about the variance of your data, the variance of the scores. Now, variance is a statistical description of a data set that tells you the size of the differences between the scores in that data set. So if you're measuring height and everyone in your sample is exactly 5'6", then the variance is basically 0. But if you have people who are 7'4", all the way down to 4 feet tall, then the variance will be much larger. So let's illustrate this concept visually. So here I've created 3 populations of dots that vary in size. So on the left is a population of dots with low variance. And you can see visually that the sizes of these dots is very similar. Not identical, there is some variation, but not very much. And so if you were to plot the distribution of the size for this low variance population, it would look something like the red curve seen here, where it's tall and narrow, and most values are close to the mean. Next, in the middle, you have a population with medium variance. And just by looking at the dots, you can see that there's more variation between them in terms of size. And you can see the corresponding blue curve below is also wider. And finally, on the right is a population with a high size variance. And you can see that visually, okay? Some dots are very, very large, and others are very, very small, and you have kind of a gradations in between. And the corresponding green curve on the bottom depicts many scores falling far above the mean and many scores falling far below the mean. All right, so that's the idea of variance, which measures the variability within some data sets. And now, how does that affect experimental studies using between subject designs? Well, it turns out that increasing or decreasing the variance can substantially affect the statistical interpretation of your results. Specifically, a high variance within your conditions can make it more difficult for you to detect any differences or changes that may occur between the conditions. All right, so let's consider the following example. Suppose you have a low variance population, and let's say you take a sample from that population, and suppose you break that sample up into 2 conditions. So treatment condition 1 and treatment condition 2. And they're both pretty similar and both have low variance in terms of the size of these dots. And now let's say that treatments 1 and 2 are different drugs designed to increase the size of the dots. Okay, and you're going to compare the effects of these drugs. So you run your experiment and you collect your data, and this is what treatment 1 looks like after the drug. And here's what treatment 2 looks like after the other drug. And suppose you find that treatment 1 had no effect, and treatment 2 increased the size by approximately 5 units. Now, before doing any stats, before worrying about significance or numbers or analysis, just looking at these two data sets visually, you can probably see that there is a difference. The dots in treatment 2 are larger compared to the dots in treatment 1. And one of the reasons that you can see this difference, the effect of your treatment, is because the variance in this data set, in this population, is low relative to the effects of your treatment. And that allows you to see the effects of that treatment. So now let's consider the following example. Same experiment, same design, same everything, except now you're dealing with this high variance population that we created. And so you have your two treatment conditions, but now you have a lot of variance within each condition. You could see that here. And so again, each one gets one of the two drugs, treatment 1 drug or treatment 2 drug. You run your experiment, and let's say the drugs work exactly the same way. Treatment 1 has no effect. The sizes remain exactly the same. Treatment 2 increases the size by approximately 5 units. And so these circles are now bigger than these. Now, again, the drug worked exactly the same way, but visually looking at these two data sets, it's probably harder to notice a difference between them, unlike the example in the previous slide. And the reason is because the variance within these two conditions is very high, and that high variance is obscuring your ability to see the effects of this treatment. So let's take another very, very similar example. So again, imagine you have two populations, population A and population B. And suppose that in population A, the individual differences are relatively small. And you can see that just by looking visually at these scores. They are all very similar to each other. So you have low variance within population A. Now, next to population A, you have population B, where the individual differences are relatively large. And again, you can see that just by visually scanning these values. There's a lot more variation in population B. All right, so we're going to perform a very similar thought experiment as we did before. So imagine you have your low variance population where the individual differences are relatively small. And suppose you have your control condition and your treatment condition. And let's say you run the experiment and you find that your treatment indeed had an effect. And the effect was approximately 10 points. Okay? So the values here are about 10 points higher than they are here. Now, if you plot these two distributions of the control condition and the treatment condition, you can clearly see that difference. It's obvious. It jumps out at you. The distributions are non-overlapping. You can see that the treatment condition had this effect. The scores are higher. And one of the reasons that you can see that difference is because the individual differences are small within these conditions, and the low variance allows you to see the effect of your treatment. And now, the same exact experiment, but using the high variance population where the individual differences are relatively large. And again, let's say you have your control condition and your treatment condition. And let's say that the treatment works exactly the same way. It increases scores by approximately 10 points. Okay? So it works exactly the same way as it did before. Now, if you plot these two distributions, the control condition and treatment condition, suddenly you can't really see that effect. I mean, you can sort of see that the blue distribution is shifted a bit more to the right than the red distribution, but it's a very subtle effect, very difficult to see. It doesn't jump out at you in the same way that it did here, even though, again, the treatment had exactly the same effect. And so what's happening is the large variance within your conditions is obscuring your ability to detect the effect of your treatment. And so, from a statistical standpoint, if you were to run your analysis on this data set, there's a good chance that this difference would come out as being significant. Conversely, if you were to run your analysis on this data set, there's a good chance that this difference would not come out significant, even though the treatment is having exactly the same effect in both cases. All right, so when talking about differences, differences between groups or between conditions are good. That's what you want. That's the effects that you are looking for, right? You're comparing two or more conditions, and you're hoping that there is a difference as a consequence of the independent variable. So differences between groups is what you are hoping to discover. And so you want to do what you can to try and increase the differences between groups, between conditions. For example, if you are studying the effects of temperature on problem solving, you probably don't want to compare 72 degrees and 73 degrees. That's a very small difference. And so if temperature is having an effect on problem solving, you probably won't notice it if you're comparing conditions that are so similar. Instead, you want to compare maybe 65 degrees and 85 degrees, and that will increase the differences between your groups if temperature is in fact having some influence on problem solving abilities. Now, conversely, when it comes to differences within groups or within group variance, ideally you want that variance to be small or smaller. Because as we saw in the previous examples, if you have very large variance within your groups, that can obscure the effect of the independent variable. All right, so variance can be a problem, especially when talking high variance within treatments. So what's to be done? How do we remedy this situation? Is there any way to lower the variance within our treatments? And the answer is yes, there are some recommendations that we'll go through. So one approach is standardizing procedures and treatment settings. So the idea is you want your participants to be treated the same way, all the participants to be treated the same way, of course, with the exception of the independent variable. So for example, if you are measuring problem-solving abilities, if you are nice to some people and rude to other people, plausibly that could affect their scores. If some people are tested in a quiet room and some people in a noisy room, that could affect their scores. And so if that's not what you're manipulating, you want to make sure that people are treated the same way. Because if they're not treated the same way, that can exacerbate the variance within your conditions. So another approach is one that we've talked about, and this is limiting individual differences by holding a variable constant or restricting that variable to some specified range. Next is random assignment and matching, which doesn't actually reduce the variance within groups, but it does help overcome or potentially mitigate the confounds that could arise from individual differences and differences between groups that are not caused by your independent variable and are therefore considered a confound. And finally, sample size. So we'll talk more about sample size in the chapter on statistics. But in general, the larger the sample size in your study, the more likely it is that the results of your analysis come out as significant and detect the presence of an effect if an effect exists. So in some ways, a very large sample size can overcome the problems that arise from high variance within your conditions. However, this is less efficient than controlling that variance through other procedures, like holding variables constant, because a very large sample size implies a lot more data collection and a lot more expenditure of resources.